But what if I told you that this replication crisis apparently also extends to other fields of science, including cancer research? And we're looking here at a paper that says a massive 8-year effort finds that much cancer research can't be replicated. So you may have heard that there's a replication crisis in science. And what that means is that when a researcher tries to repeat or replicate the work of another scientific researcher that's been published, that means that that researcher has trouble or is unable to get the same results. This is noted to be more prevalent in some fields than others, and for instance, I'm looking at a source here from replicationindex.com that says, in fact, a moderator analysis suggested that the replication rate in cognitive psychology is 50%, while the replication rate in social psychology is only 25%. That means that if researchers tried to repeat 4 research studies that had been published in a peer-reviewed journal, only one of those actually repeated, which argues that potentially those other 3 studies were potentially a waste of time. Fortunately, there are some caveats to this. So it says the replicated studies were also limited to a single year, 2008, in 3 journals, and it's possible that the replication rate has increased since 2008 or could be higher in other journals. But it's still very unnerving, and it tends to reduce your confidence in psychology data in general. But what if I told you that this replication crisis apparently also extends to other fields of science, including cancer research? And we're looking here at a paper that says a massive 8-year effort finds that much cancer research can't be replicated. I also want to say, from my perspective as a scientific researcher, replication issues can be incredibly demoralizing and frustrating. As I explored in a recent video about science fraud, there can be a number of different reasons for failure to replicate, including sometimes that scientific data was actually faked. But as we'll discover from the results from this 8-year study, a lot of the time it has nothing to do with fraud, but there are other reasons why, in particular, cancer research is really hard to replicate. So obviously when it comes to cancer research, the importance of repeatability or replication are even more important than it might be for other fields. And to illustrate that, I wanted to take just a brief detour to look at this study from sciencenews.org that talks about a drug known as EPO. It says, in animal studies and early clinical trials, the drug appeared to counteract anemia caused by radiation and chemotherapy. It therefore had the potential to spare patients the need for blood transfusions during radiation and chemotherapy. They also had evidence that it might increase the radiation's tumor-killing power. But it says when doctors started giving EPO or related drugs to large numbers of cancer patients in clinical trials, it looked like deaths actually increased. Physicians were concerned and some stopped their studies early. At the same time, laboratory researchers were collecting evidence that EPO might be feeding rather than fighting tumors. So that just goes to show that the replication crisis when it comes to cancer research is incredibly important. We have to get to the bottom of this. So let's take a look at the actual results from the Reproducibility Project. So as I said before, this was an 8-year effort to replicate experiments from high-impact cancer biology papers published between 2010 and 2012. And what the researchers who carried this study out tried to do is basically prepare replications of 193 experiments from 53 papers, which is a monumental amount of work, especially when you consider the fact that, for me, the first scientific paper I ever wrote took me a year of part-time work and a year of full-time work. So it took 1.5 years of full-time work. Granted, it's a lot easier, presumably, to follow the instructions that somebody else put forward rather than to sort of invent your own recipe, but nonetheless, 193 experiments, and these are biomedical studies. This is an incredibly huge amount of work. So here's some key data from the overview. So first of all, they found that only 2% of the studies actually published their data openly, and 0% of protocols were completely described. In other words, none of the scientific protocols that were written out in the papers were actually enough to totally repeat what they did. There were always missing details that prevented them from fully repeating the work. In addition, a lot of the materials or reagents that were used in the specific studies were rare enough that they actually had to ask for key reagents of the initial researchers. So they actually had to follow back up with the original authors and not only ask them for the missing details in their protocols, but actually ask them in some cases for key reagents. But that's fine as long as they're willing to share the reagents, right? But it says that 32%, the original authors, were not helpful or were unresponsive. And in fact, the larger write-up of this study actually said that among these 32% of original authors who were not helpful, many of them were actually hostile. It's not clear why they were hostile. It's possible that they felt threatened. Academic research is very cutthroat, and so maybe they felt like if these people were able to repeat their work, then they would be able to do some research that would kind of get a jump on whatever they were doing and publish some key result before they were able to. But I think this is actually an extremely unforgivable attitude, especially when you consider the fact that the vast majority of this research is going to be in part or in full funded by taxpayer dollars. Ultimately, they found that only 50 of the original 193 experiments were actually completed and generated data. They also found that the effect sizes were 85% smaller on average than the original findings, which that means that, for instance, if the original paper claimed to kill a certain amount of cancer cells using their therapy, then the repeated researcher found that the amount of cancer cells that were killed was actually lower than that, maybe 85% lower. And they also found that original positive results were half as likely to replicate successfully than original null results. And so a positive result would be to say that, well, we found that this specific therapeutic was effective in killing cancer cells. And the null result would be to say that we found that this specific proposed therapeutic was not successful in killing cancer cells more than, say, just an average amount. The fact that the positive results were less likely to replicate successfully argues that most likely the researchers were maybe, without intending to, kind of, you could say, putting their thumb on the scale, kind of maybe selecting for or rounding up. Again, probably not intentionally, but sometimes when you're doing scientific research and you really want to see a specific result, you're just going to introduce unintentional bias in your measurements themselves. And so the fact that, statistically speaking, when they looked at this research overall, they found that the positive results were less likely to replicate successfully than the negative results, that implies that amongst all these researchers, the presence of bias was actually a statistically real effect in terms of influencing what they found as the actual findings of the report. So what should we take away from this? That cancer research is a waste of time or cancer research is fraudulent? I don't think that's the right conclusion. In fact, you just have to remember that the stakes are so incredibly high with cancer research that we can't afford to throw out the baby with the bathwater. And I think that the original report about this research provides some really good conclusions as to what we should be taking away from it. They say, first and most obviously, preclinical studies, so these biomedical studies, should never be interpreted in isolation from other theory and evidence. Instead, decision makers, that's especially the people who are going to decide whether or not a preclinical research study should actually be translated into a clinical study, which is involving real patients. It says that instead, decision makers should actively seek corroboratory and disconfirmatory evidence and interpret any claim in a preclinical study against what was known previously. So they shouldn't take sort of some fancy, exciting research from a new cancer study just kind of in isolation and say, wow, this is great, we found a cure. They need to really be thorough about comparing this to the known body of literature. Second, as a general rule, positive preclinical publications should be understood in light of their role as providing exploratory evidence for future clinical work. And I think this is the most critical part. It says that we need to maintain strong mechanisms for subjecting clinical hypotheses to a retest before they are advanced into clinical practice. And I think it makes a lot of sense, actually. Maybe if the rule should be not only that the original researchers should be able to repeat their work, but that maybe another group of researchers should be hired just to see if they can repeat the findings from the original study. I think that would be an incredibly valuable innovation to this sort of therapy development process. The other main thing that I think needs to happen, and this is purely my opinion, is that these two numbers here need to change. This 0% of protocols completely described absolutely needs to change. And this 32% of experiments where the original author were not helpful or unresponsive absolutely needs to change. This speaks to an incredibly suboptimal research culture in science overall. The fact that 0% of protocols completely described, I can actually attest to this, that when I was in graduate school, there were conversations that were had, specifically among grad students, some of whom were taking their lead from the professors and some of whom were not, that were along the lines of saying, well, when we write up the experimental protocol or the methods and materials section of our research papers, we don't want to include all the details because it might make it too easy for someone else to replicate. And the reason why they thought this was a threat was because in academia, there's this publish or perish mentality, which is to say that if you don't publish cutting edge research that other people are not doing, then you're not going to have a successful academic research career. And so that means that if you write your experimental protocols too thoroughly, then it might make it too easy for someone to replicate your work. But actually, collectively, as a society who's funding science and wants the world to change for the better, we should actually be maximizing the ease with which researchers are able to replicate each other's work. I think the thing that made me even more upset than that was this 32% of experiments where the original authors were not helpful. That's just absolutely unacceptable. I wish that they would publish the names of these researchers, and I think, honestly, that they should be publicly ridiculed. That they should be shamed for this, because that's just unacceptable. That's putting your own interests as a career-oriented professional above the interests of the collective when it comes to scientific advancement. And that is especially unforgivable when you consider the fact that, by and large, these researchers are being funded by taxpayer dollars. So that absolutely needs to change. I think instead of being threatened by someone repeating your work, if you're really thinking about it right, you should be honored and excited. And I have to say, actually, that the first paper I was talking about before, the first paper that I ever published, and it's not in cancer research, it was in polymer chemistry. There was a really great moment I had, which is when someone actually didn't quite replicate my work. They did something very, very similar. And when they published their paper, they actually included a figure where they had my synthetic scheme and then somebody else's synthetic scheme and then their synthetic scheme, and they showed the differences between them and maybe made an argument about why theirs was better. But that made me feel so incredibly validated because someone had looked at my research, they had considered it worth thinking about and actually emulating and maybe making an improvement on. And I felt like I had actually been part of a scientific conversation, and that maybe in some very, very small way we had together, that is me and the person who replicated my work, made the world a little bit better. Maybe turned the lights on in the room just a little bit more so we can see and understand the natural world better, which might imply improved technologies in the future. That was an amazing feeling, and I think these 32% of original authors who are not helpful or unresponsive, if they could just take that point of view, I think that they would have a much more fulfilling scientific experience. So overall, I think this is an amazing study. I'm really glad they did this. This is a huge effort, an 8-year effort to replicate these experiments. We're talking trying to repeat 193 experiments from 53 papers. I hope that the field of cancer research and just science in general can take note of this and treat this as an opportunity to really change the way we do science. Well, I hope you enjoyed this video. If you found it interesting or helpful in any way, I would greatly appreciate it if you would hit like. Thanks so much for watching, and I hope you'll join me again soon in the future. Bye.